Hey everyone, Darwin's God here. Last time we looked at the idea of junk and how important this idea is in evolutionary thought. When evolutionists see a junky design, they interpret it as very powerful evidence for evolution. Now we also saw that this idea transcends evolution and goes all the way back to the ancient Epicureans 2,000 years ago. It's a good example of the link between Epicureanism and evolution. And it's a good example of how modern evolutionary thought isn't so modern after all. So this time I'm going to take a closer look at this whole argument. What we're going to see is that it falls apart very badly. It's not powerful evidence for evolution, it's powerful scientific evidence against evolution. So here we go. Okay, so one of the examples we looked at last time was from this book, The Blind Watchmaker, written by Richard Dawkins. Dawkins, a leading evolutionist. He was a life science professor at some of the top universities in the world, a very smart guy, top evolutionist, and he made this argument that the vertebrate photocells are wired in backwards. Now, what do we mean by that? Here's a simplified look at a squid eye and a human eye, and it shows that the squid does not have a blind spot, but the human does. Let's zoom in on that here. So look on the left here, the squid retina, this is the back of the eye, the light's coming in from the left, and the photocells are lined up in kind of a layer, and then the neurons behind them, or wires carrying the signals to the brain, come out the back. They go over to this hole in the retina, that's the optic nerve going off to the brain, okay? That's all well and good. For the human, you don't have it that way. The photocells are lined up in a layer, but the neurons come out the front side and are layered on top of the photocells in the way of the oncoming light, and then come over together and join together in this optic nerve going through the hole in the retina. So you can see in that design it causes a spot here where there are no photocells, so you have a blind spot. So back to this quote here, each vertebrate photocell is in effect wired in backwards, says Dawkins. So can you feel the weight of the argument here? Let's see a video on this just to give you a better idea.
And interestingly, this photon of light is actually going to go through these cells until it hits these cones right here and these rods.
Okay, did you catch that? This is an image of the human retina. It's a little more detailed than that other graphic we just looked at. The other graphic showed the photocells only, this blue layer, but there's also a couple of other layers that help out, the green layer and the red layer of cells. So the light has to pass through not only the neurons that are carrying the signal to the brain, but also this green layer of cells and red layer of cells before it can get to the photoreceptor cells. In fact, the part of the photoreceptor cell that detects the light is at the very back. There's the kind of front half that does regular cell jobs that's in the way also. So the light has to pass through all this material before it gets to the actual business end of detecting the light in these photoreceptor cells. So back to our graphic here, you can see that the squid does not have a blind spot. The human does have a blind spot. And you can kind of feel the weight of this argument that Dawkins is making. It seems that the human eye is junky. It's a junky design. The photocells are wired in backwards. Okay, I'm going to look at three problems with this argument: convergence, cascade, and conversion. 3 C's here. So convergence, what's that? Well, back to our graphic that we just looked at, the squid and the human eyes. Yes, there is this difference of one has a blind spot and one doesn't, but otherwise they're amazingly similar. Too similar for evolutionary theory. Remember, evolution works off of random mutations. You have random mutations over a long period of time just kind of happening to put together a design. The squid and the human are on very distant parts of the evolutionary tree. Their common ancestor would be way back early in time, early evolution. Did that early ancestor have an eye that the squid and the human would have inherited? Was this eye present way back then? No, it was not. What this means is that the squid vision system and the human vision system had to have evolved independently, and this is a problem. This is called convergence. The sequence of mutations required to put together the human eye is very low probability. That has to happen twice now. Once in the squid lineage, once in the human lineage. These had to have happened independently. Therefore, the probability is now squared. It is a insanely low probability. It's not going to happen. The entropic barrier is way too large for this to happen. This is called convergence. Darwin was vaguely aware of the problem. It had been raised, and he was very concerned about it. Today, it is far more than Darwin could have dreamed of. It is ubiquitous in biology, and this is an example of it where you have these strikingly similar designs in distant species. Now, evolutionists often will appeal to natural selection to do the heavy lifting. Yes, it all seems very improbable, but natural selection is going to make it happen. It's going to pull out those low probability mutations, that low probability sequence of mutations. You have similar environments, similar ecological niches, and so forth. You can expect similar results. Well, no, that argument is highly problematic. I'll just point out a couple of problems. One, natural selection does not induce mutations to occur. Natural selection does not induce the right mutations to occur. All it can do is kill off the bad ones that screw things up. Secondly, you could hardly think of a more different environment than the human and the squid. These are hardly a similar ecological niches that they exist in. So the whole natural selection argument fails, and the science is contradicting the theory once again. This is a problem for evolution, the convergence problem here. This is called convergence when you have similar designs that happened repeatedly. Darwin was aware of this vaguely. The problem had come up, and he was very concerned about it. Today it is much, much worse, and this is one example of that. Okay, next let's move on to Cascade, the Cascade problem. Back to our graphic here, our more detailed graphic. So what's happening here? The light's coming in from the left. It's going through the green layer and the red layer and these blue cells until it gets to the back where you have these small organic molecules called retinols that when light hits them, they change shape. When they change shape, they influence a protein that they are attached to. That's event A. Let's call that event A. When that protein is influenced, it influences another protein. Let's call that event B. When event B occurs, you then have event C occurring. When event C occurs, it influences another event. Let's call it event D. And so forth and so on. A series of events occur leading up to the closure of sodium channels in the membrane. So this outer membrane of these photoreceptor cells have a large population of sodium channels. These are very complicated protein machines that have a channel that just allows sodium through. It doesn't allow potassium or other ions. It allows only sodium through. And normally they are open and sodium ions are streaming into these photoreceptor cells, maintaining a certain voltage across the membrane. When those channels are closed down, that stops the flow of the sodium ions. When you stop the flow of the sodium ions into these photoreceptor cells, that changes the voltage across the membrane. That event sets up a different set of events which ultimately ends, it influences the red layer, which influences the green layer, and may end up activating an electrical signal in the neuron, the nerve cell, going to the brain. The red layer and the green layer do kind of, we might think of as post-processing on the signal, and they're gathering these signals from multiple photoreceptor cells. So to recap, you've got event A, event B, event C, event D, et cetera, leading up to a signal being sent to the brain. It's a very, very complicated process. So again, the science is contradicting the theory. The setup here, the sequence is far too complex, far too finely tuned. The entropic barrier is enormous. The random mutations for random mutations to find this design is simply 10 to the minus thousands and thousands. This is not an argument from incredulity. This is an argument from science. The science is contradicting the theory. What we know from today's science flatly contradicts the theory of evolution. Now, there's another interesting aspect of this thing. Why is it called Cascade? So I said that you have event A leading to event B, C, and D, and so forth. What's interesting is that it's not simply a case of one event triggering another event. It's actually a case of one event triggering a large number of other events. So one event A triggers maybe hundreds or thousands of event Bs. And one event B triggers a large number of Cs, maybe again hundreds or thousands. And the same thing for D. And why this is important is because one photon or just a couple of photons entering these photoreceptor cells normally really couldn't get the job done. There's no way a single photon or a few photons is going to be enough to trigger the sort of electrical change we need to have happen. But because this is a cascade, you have an exponential rise. One or a few photons can create a large number of events by the time you get to C and D, and eventually the number of sodium channels closing in this cell membrane is enormous. And amazingly enough, humans have an incredibly sensitive vision system. This was discovered in the last century, that we can actually detect one or a few photons. It was amazing. That discovery was quite amazing. So that too contradicts the theory of evolution. It's far too finely tuned for this cascade sequence to be set up like this. Is very finely tuned and contradicts the theory of evolution, which of course states that random mutations would have to set all this up. So I've pointed out a couple of things amounting to incredible fine-tuning that contradict the theory of evolution on this design. But there's still Dawkins's complaint. And by the way, it's not just Richard Dawkins. This is a very common argument. As I said at the beginning, junk is a key concept in the evolutionary thought. So you've still got this problem of this kind of backward design, right? These photocells are at the back of this process here, and the light has to go through all of this material to get there. Isn't this? And then you have the resulting blind spot. It all seems to be kind of a junky design, right? Well, no, again, that's not the case. The science, despite evolutionary thought, despite evolutionists, the science contradicts the theory. And let me show you just a couple of example papers here. Here's a paper that explains having the photoreceptors at the back of the retina is not a design constraint. It's a design feature. Okay, the idea that the vertebrate eye, like a traditional front illuminated camera, might have been improved somehow if it had only been able to orient its wiring behind the photoreceptor layer, like a cephalopod or a squid, is folly. Okay, folly. So it's not a problem, it's not a bug, it's a feature. Here's another paper. The retina is not just the simple detector and neural image processor as believed until today. This is a professor out of Israel. Quote, its optical structure is optimized for our visual purposes. Optimized. Here's a more recent paper out of just last month here. Cone mitochondria act as microlenses to enhance light delivery. Now the cone mitochondria, what that's talking about is the mitochondria organelles in the cone photoreceptor cells. So remember, the light had to go through the green layer and the red layer. It also had to go through half of those photoreceptor cells. What this paper is showing is that in that half of the photoreceptor cells, there are these organelles called mitochondria that you will recall from your high school biology class. There's a lot of mitochondria in those cells, and they themselves are assisting with the transmission of the light. It's a fascinating finding. So while evolutionists have been making these kind of vague arguments about junk, scientists have actually been discovering the function that's going on, and there's a lot of them. It's very complicated, by the way. Okay, so now on to our last C, conversion. What's going on here? You have the eye, you have the brain. The eye is detecting the light, sending the information to the brain in the right, upper right there. Well, what's actually happening is the light is coming into the eye, hitting the photoreceptor cells, and then that's being converted to electricity. That electricity is sent to the brain, and you have an image. That middle part is important, electricity. There is no optical information. It's not like there's, fiber optics running to the brain. And there are millions of these nerve cells in this optic nerve. So you've got kind of think of it as a wire bundle, each of the wires sending electrical information to the brain. Here is a typical kind of figure that you might find on the internet. It shows an image entering the eye, impinging on the back of the retina, and then you have the optic nerve, and then you have the image being projected in the brain. Well, that's misleading because there is no image being projected onto the brain. There is no color or image being projected. This is not an optic fiber, it's an electrical fiber. Don't think of this as a projection system, so it's a little misleading here. So back to our figure, electricity is being sent to the brain. Now you're looking at this right now and you're seeing an image. How is that electricity being converted into an image? How are you seeing the blue here around this guy's head? How are you seeing the red in these letters? How are you seeing the image at all? There's no scientific explanation for this. How the electricity is converted into a conscious sensory perception that you have. There's no scientific explanation. There's no natural laws that explain that conversion. So look at this electricity, then you got the brain, then you got the image. That step right there, electricity to image. There's no explanation for how that works in today's science. Maybe that will be discovered someday. Today we have no explanation for that. So the science does not support the theory of evolution. You have no natural laws to do this. There's nothing for natural selection or random mutations to do here. They could not create this. Now again, maybe that'll be discovered sometime in the future. I'm not talking about the future. I'm talking about now, today. What we know, what the science is telling us, is this is impossible for evolution to do. There's no natural laws that evolution could work through to do this. The science is contradicting the theory. You cannot get a conscious sensory perception from a bundle of electrical signals by natural law. Okay. Let me expand and expound on this just a little bit, going back to our figure here, our more detailed figure. Again, the blue layer here at the back are the photocells. There are two types, rods and cones. The rods do grayscale, the cones do color. You might wonder, how do they do color, because there are different colors? If there's a bunch of cones, how are they going to get all the different colors? Well, it turns out there are subtypes of these cone photoreceptor cells. S, M, and L are three types. The S stands for short, and M medium, L long. So cone cells that are S-type have a retinal in there that change conformation when hit by wavelengths that are shorter, around 450 nanometers. Longer wavelength light, say up around 600, the short cone cells are unlikely to activate because that little organic molecule, the retinol, will not change conformation. So it's a different retinol in there that's sensitive to different wavelengths. In the L type, the retinol is more sensitive to the longer wavelength. And then in the M, you're in the middle somewhere. So this is a nice graphic that was drawn, because overlaid on this graphic are the colors that we're used to, right? So if you ask someone, well, what does the S-type cone photoreceptor cell detect? They'll say, oh, blue. And what does the M-type do? Well, that's green. What does the L-type do? Well, that's red. But they don't actually detect blue or green or red light. What they detect is 450 nanometers or 550 nanometers or 650 nanometers and so forth. In other words, the light coming into your eye is not red. It's not green. It's not blue. It's not violet. It's not aqua. It doesn't carry any of those colors. Those colors are not there. it's hard for us to kind of wrap our heads around that. It's just different wavelengths of light entering your eye. The cones, different cones are activated by those different wavelengths of light, and then they will send their signals to the brain. Back to our image here, the brain then is going to get this wire wrap, these millions of signals coming in, and it will interpret those signals. It puts the colors on. The colors are not on the light to begin with. So the medium is not the message, rather beauty is in the eye of the beholder. We are putting those colors onto the message. We are interpreting the light in our minds. When you convert the electricity to the image, again, there's no natural law to explain this. That's where the colors are introduced. So those colors that are on that image here are simply reflecting how our minds work and adding those colors to that. Now, how could this all have evolved? Again, it's more of the same problem. The science is contradicting the theory. Somehow, the mind must be interpreting those colors. It must know, aha, this signal here is coming from a blue cone, right? This one over here is coming from an M cone or an L cone. I'm going to put some colors on these. How would the brain know that? How, under evolutionary theory, how would the brain know to attach these colors? There are millions of these neurons coming into the brain with their electrical signals. How would it know that this set is from the S type of cone and this set is from the M type of cone, and so forth? Even if you were somehow able to evolve this, we looked at the problem of this evolving in the first place, the retina and so forth. Even if you were able to evolve that, the electrical signals do not convey that color information. So not only are there no known natural laws by which this transformation from electricity to an image could occur, and so therefore there's nothing for natural selection and random mutations to even do here, but even if that was to be discovered, the fine-tuning here, the complexity of matching those up from the brain side and the photoreceptor side is astronomically unlikely to happen. It's an entropic barrier that is astronomical, and random mutations is simply not going to be able to put that together. And again, natural selection doesn't save the day, because natural selection cannot induce mutations to occur. It cannot induce certain types of mutations to occur. So we've looked at three problems here, the three C's: convergence, cascade, and conversion. Each one of these problems contradicts the theory of evolution. The science is contradicting the theory of evolution. And when we look at what evolutionists are saying, again, it's not just Richard Dawkins. This is a very common argument. Hey, the photocells are wired in backwards. This is really a non-scientific argument. It's actually a science stopper. While evolutionists are busy ascribing junk and identifying junk in biology, scientists are finding all kinds of interesting and fascinating function and design information in biology. Very interesting world, in spite of what evolutionists are saying. And so the evolutionists, it's not that they're dumb. They certainly aren't. These are very smart people. But the wisdom that they have is failing. And just remember this verse here, I will destroy the wisdom of the wise. Keep that in mind here, because that's very much at play here. I will destroy the wisdom of the wise.